Thank you for the introduction. I should make the title a bit shorter next time. Hi, everyone. This is really with such a pleasure to be able to present to you this highly collaborative work on ARHGAP19 loss of function that sparked my curiosity in RHO-A regulation in CMT. So thank you to the organizers for the opportunity to present. I'm a fundamental scientist at Queen's Square UCL, and I will take you through the story of ARHGAP19. But today I will really focus on the mechanisms and the drug screen that I'm currently working on in my first postdoctoral role. Now, you might have seen me present this slide before, but just really briefly, the index patient, she came from a consanguineous family, and at the age of 9 years old, she presented with frequent falls. She walks with, She walks with foot drop and has distal lower limb wasting and pes cavus. After next generation sequencing, we found a biallelic variant in ARHGAP19 in her that later segregated within her family. Now, admittedly, this was quite a few years ago, and now up to date, we've collected 25 patients from 20 families from different ancestral backgrounds with biallelic variants in ARHGAP19. Over 70% of the patients came from consanguineous parents, and the mean age of onset was about 10 years old, but it really ranged from infancy to about 30 years old in one of the patients. The presenting symptom was motor problem at lower limbs, and over 60% had some sort of sensory involvement. The neuropathy was mild to moderate and motor predominant. The patients, they had various foot deformities such as pes cavus, flat feet, or hammer toes. Now, moving on to the mechanism, ARHGAP19 is a raw GTPase activating protein of just under 500 amino acids in length. It has this evolutionary conserved GTPase activating protein domain or GAP domain. And at its C-terminus, it has some residues that can be phosphorylated, and they're very important for its subcellular localization. In our patients, we've found 16 different biallelic variants in ARHGAP19. 9 of them are within the GAP domain, and 7 of them are outside of the GAP domain. But what does ARHGAP19 actually do? Well, I've mentioned to you already that it's a GTPase activating protein. It has activity towards RHO-A, where it stimulates its low intrinsic GTPase activity. RHO proteins, they are GTPase proteins. They act as molecular switches. And they basically cycle through inactive GDP-bound state to active GTP-bound state. When they're in their active form, they can go on to activating many different effectors, and RHO-A can activate effectors with role in actin organization, cell cycle controls, and migration regulation, and many others. RHO-A regulation is really tightly regulated by many regulatory proteins, GDIs, GAFs, and those GAPs that we're talking about today. Now, knowing that ARHGAP19 has a GAP activity towards RHO-A, where it negatively regulates the pathway, we wanted to see whether the variants that we see in our patients within the functional GAP domain, if they have any consequence on RHO-A regulation. We can do it through in vitro GAP activity assay, where we measure the release of inorganic phosphate at GTP hydrolysis. And indeed, all the variants that we found in our patients that we tested, they have near complete loss of GAP activity towards RHO-A, which is really quite striking when compared to the wild type. Now, we went to model the loss of function with 2 animal models, and starting with Drosophila melanogaster CRISPR-Cas9 global knockout of the gene ortholog RHGAP54D. From the available data sets, we saw sparse expression of RHGAP54D in the fly. And from our own staining, we saw the protein expression in subperineural and perineural glia in adult fly brain and in some cells of thoracic ganglion. Now, because the patients, they have problems with walking, we wanted to see whether the knockout of RHGAP54D in the fly will similarly impair the fly locomotion. We can do it through Drosophila activity monitor, where we put flies in glass tubes, 1 fly per tube, and they walk back and forth. When they break this infrared beam in the middle, this is counted as movement. And we do it in 24 hours, in 12 hours light and 12 hours period, so we can also look at their activity patterns. And indeed, when we looked at their gross locomotion, we saw that the knockout flies had significantly lower locomotion than the controls. And also looking at their activity pattern, the peak activity is at the change of light, and this was significantly impaired. We were also able to look at the larvae of these flies, where they had similar phenotype and they moved less. But also looking at their neuromuscular junctions and particularly looking at the neurotransmitter storing boutons, we saw that the number of boutons was much slower, but also they had a larger surface area, which was previously also seen in cytoskeleton-related mutations. We also saw in those knockout flies that the axonal length was shorter. We also went to model the knockout of ARHGAP19 ortholog in the zebrafish, and there we saw similar phenotype where the zebrafish moved slower, and also there was less movement in them. But what I wanted to draw your attention to here are actually the axons. So looking at the axons of the knockout zebrafish, we saw increased axonal branching and decreased axonal length. And we were able to look at patient-derived iPSC motor neurons as well. We were able to do it in 2 patient lines compared to 2 controls. And here we saw similar phenotype that we saw that the neuroid branching of the patient was significantly higher than in the controls. We didn't see any phenotypes when it came to neuroid length. We looked at the protein expression in those iPSC-derived motor neurons, and ARHGAP19 was less expressed than in the controls. This work is now in press with JCI, and with this I would like to move on to my current part of the project, which is looking at the therapeutic targets, utilizing my Drosophila melanogaster as a model organism. Now, Drosophila is a fantastic model organism and a great entry point for a drug screen because of the large number of animals, very quick lifespan, and they're relatively easy to house. Also, we can look at many different time points, and for my drug screen, I'm using adult flies straight after eclosion, so when they emerge from the pupal case. My drug repurposing screen setup is really quite simple. I take FDA-approved drug library or DMSO as control. I dissolve these in fly food, and I let my flies feed for 48 hours. And then I put them in the Drosophila activity monitor, as I explained before. Now, these flies are still either on the drug or on a DMSO control. And I do it with an aim to look at any candidate drugs that might rescue the Drosophila locomotion phenotype. Now, because I know that ARHGAP19 has a gap activity towards RhoA, I thought that ARHGAP19 loss of function could cause overactivity of RhoA, or also of ROC, if it's indeed an effector in this pathway. So I went to feed my flies with RhoA and ROC inhibitors. And for RHO inhibitors, I didn't see any significant changes in the fly locomotion or in the activity pattern. With the ROC inhibitors, well, I didn't really see any changes either here. And now this could be because I missed the critical period of the drug functioning, because the dosage was incorrect in this model organism, or simply because these are not relevant here. So I went to expand my drug screen with an unbiased drug screen, still in adult flies. And here I'm happy to report this is going well. In pink, you can see my DMSO control, and you can see some compounds on the right. I'm interested in 2. However, I will have to leave you on a to be continued here. I'm really sorry about that, simply because I would like to validate them with patient tissue and hopefully with the zebrafish model. Also, in my future pipeline, I would like to go back to the RHO and ROC inhibitors and use them at different concentrations and time points. With this, before I move to my thank you, this is my contact. Please, if you have any ARHGAP19 patients, if you have any tissues, please do contact me. And I'm very fortunate because I have many funders to thank. I would like to thank my current funder who is allowing me to continue this work, UCLH, BRC, previous funder, ICGNMD, and others. And I'm very lucky because I have many colleagues and collaborators who helped with this work. But I would like to especially thank Stephanie Eftemu for co-leading the first part of the study with me, Chris Record for the clinical help, Mary Riley, Andrea Cortez, and Henry Holden for supervision and for mentorship, James Jepson for allowing me to use his Drosophila lab. I would like to also thank my son who is very heavily involved in my work, and then all the other collaborating clinicians and scientists, and of course the patients and families for taking part in the study. With this, I thank you for your attention. I'll take any questions, comments, or advice, please.